Now we'll transition to the last subsection of biochemistry, the metabolism section. Metabolism encompasses the various processes that contribute to cell energy requirements, structure, and waste removal. This involves the creation and interchange between four classes of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. It can be useful to group metabolic reactions by the site in which they occur. Most metabolic reactions occur in either the mitochondria or the cytoplasm, although some occur in both. Oxidation of fatty acids, production of acetyl-CoA, the TCA cycle, and oxidative phosphorylation all occur in the mitochondria. Glycolysis, fatty acid synthesis, the hexose monophosphate shunt, protein synthesis, which occurs in the rough ER, and steroid synthesis, which occurs in the smooth endoplasmic reticulum, all occur in the cytoplasm. And finally, you can use the mnemonic HUGS take 2 to remember which processes take place in both the mitochondria and the cytoplasm, which are heme synthesis, the urea cycle, and gluconeogenesis. It can be useful to know the meaning of an enzyme name, since this often describes its function, so you don't have to remember it separately. I'll walk you through the list of commonly used enzyme descriptors. A kinase is an enzyme that uses ATP to add a high-energy phosphate group to a substrate. For example, phosphofructokinase phosphorylates fructose 6-phosphate in glycolysis. A phosphorylase is an enzyme that adds an inorganic phosphate group onto a substrate without the use of ATP, which is what differentiates it from a kinase. For example, glycogen phosphorylase cleaves the 1,4-glycosidic bonds in glycogen to release glucose 1-phosphate by adding an inorganic phosphate to it. A phosphatase sounds similar to phosphorylase, but actually does the opposite. It removes a phosphate group from a substrate. An example is fructose 1,6-bisphosphatase, which converts fructose 1,6-bisphosphate to fructose 6-phosphate in gluconeogenesis. A dehydrogenase is an enzyme that oxidizes a substrate. This means that hydrogens or electrons are removed from the substrate. An example of this is pyruvate dehydrogenase, which converts pyruvate to acetyl coenzyme A. And lastly, a carboxylase is an enzyme that adds one carbon carboxyl group with the help of biotin. For example, pyruvate carboxylase converts pyruvate to oxaloacetate. It's important to know the rate-determining enzymes in metabolic processes. These are the most commonly tested since they determine the rate at which a process occurs. The rate-determining enzymes are also the ones most commonly targeted by drugs, because slowing them down or speeding them up will definitely have an effect, which might not be the case with non-rate-limiting enzymes. It'll definitely help you in your test to memorize which enzyme is rate-limiting for which process, and also what the role of that enzyme is in that process. For example, HMG-CoA reductase is the rate-limiting step in cholesterol synthesis. Do you remember which drug is used to inhibit this enzyme? That would be the statin class of drugs, which are used to treat hypercholesterolemia. We'll cover each of these in the next group of slides, so I won't cover them all now, but you may want to go back and review these when we're finished. This diagram may seem intimidating, but it's just meant to show you how all the metabolic processes we're about to go over are truly interconnected. Carbohydrate metabolism is connected to protein metabolism, lipid metabolism, and nucleic acid synthesis. Steps that require biotin, which are usually steps that require addition of 1 carbon units, are marked with a B. For example, pyruvate has 3 carbons and oxaloacetate has 4 carbons, or propionyl-CoA has 3 carbons, while methylmalonyl-CoA has 4 carbons. So these require biotin. Steps that require thiamine are marked with a T. And starred enzymes are irreversible steps, which are important parts of regulation. Once you feel comfortable with the different smaller pathways that make up this big pathway, which we'll discuss in the next few slides, it may be helpful to look back and see how it all fits together. This is also a great page in the book to take notes in from other parts of the chapter, or even from other chapters, since there are more ins and outs to this diagram than are shown here. For example, 1,3-bisphosphoglycerate is used to make 2,3-bisphosphoglycerate, which is used to reduce the affinity of hemoglobin for oxygen, and will be discussed more in the hemonc chapter. Also, fatty acids are broken down into acyl-CoA, which are then transported via the carnitine shuttle to the mitochondria, in which they're converted back into acetyl-CoA. As you study for your Step 1, you'll probably find lots of things to add to this page, and having lots of important stuff in one place can make it easier to study. We'll start to deconstruct that elaborate metabolic diagram by talking about one of the most important processes in carbohydrate metabolism, glycolysis. This is the first step to break down glucose to make ATP. Aerobic metabolism takes place in the cytoplasm and the mitochondria, where oxygen is the final electron acceptor. Aerobic metabolism of glucose can produce 32 ATP molecules via the malate aspartate shuttle present in the heart and liver. This shuttle transports NADH electrons gathered in the cytoplasm to the mitochondrial NADH acceptor, which can produce 3 molecules of ATP per NADH. Only 30 molecules of ATP are produced via the glycerol 3-phosphate shuttle, which is found in muscle. This shuttle transports NADH electrons gathered in the cytoplasm to the mitochondrial FADH2 acceptor, which can only produce 2 molecules of ATP per NADH. Anaerobic metabolism takes place only in the cytoplasm and does not require oxygen. It produces only 2 net ATP per molecule of glucose. ATP, which is shown here, can be hydrolyzed in otherwise energetically unfavorable reactions that would not occur spontaneously to provide the necessary energy for the reaction to occur. There are several molecules that carry functional groups that are transferred during metabolic reactions that we'll talk about soon. It can be useful to know which functional groups each activated carrier is associated with, since this can help you determine the outcome of each metabolic reaction. ATP carries 3 phosphoryl groups. It can use these to phosphorylate proteins. NADH, shown here, as well as NADPH and FADH2, carry electrons. Coenzyme A, shown here, and lipoamide carry acyl groups. Biotin carries carboxyl groups, which are used in many reactions that involve adding one carbon to something, such as converting pyruvate to oxaloacetate. Tetrahydrofolates also carry 1 carbon units, and these are mostly used for nucleotide synthesis. S-adenosylmethionine, or SAM, can carry methyl groups, and thiamine pyrophosphate, which is the active form of vitamin B1, carries aldehyde groups. In the vitamin section, we talked about the most common electron acceptors, which are the nicotinamides, NAD and NADP, and FAD, which is a flavin nucleotide. These molecules accept electrons through various redox reactions, and then transfer electrons in subsequent reactions, such as in the electron transport chain, to either make ATP or to build a higher energy molecule. NAD is mostly used for catabolic processes that break down organic molecules, and the electrons are carried in reducing equivalents as NADH. NADPH is mostly used in anabolic reactions. NADPH can be created using the hexose monophosphate shunt, which we'll talk about in a few slides. It's mostly used in anabolic processes, such as synthesis of steroids and fatty acids. It's also used during the respiratory burst to create oxygen radicals that damage bacteria, and is used in the P450 system in the liver, and by the enzyme glutathione reductase, whose function is to reduce glutathione. Now I'll talk about the first step of glycolysis, which is the metabolic process that breaks down glucose to form ATP. Its first step is phosphorylation of glucose, which creates glucose 6-phosphate by using 1 ATP. This reaction can be catalyzed by either hexokinase or glucokinase. The difference between these two enzymes lies in their locations and their enzyme kinetics. Hexokinase is found in every cell type. It has a high affinity for glucose, however has a low maximal reaction rate. This means the concentration of glucose needed to occupy the enzymes is relatively low, but the speed at which the reaction occurs is also relatively low. Hexokinase is not affected by insulin, but does receive negative feedback from high amounts of glucose 6-phosphate. So basically, when the cell has low glucose 6-phosphate, hexokinase will catalyze any glucose it finds to glucose 6-phosphate at a low rate. However, if there's a large amount of glucose in the blood, hexokinase won't be able to keep up because it has a low capacity. Glucokinase, on the other hand, is only found in the liver and beta cells of the pancreas. It has a low affinity for glucose, but a high capacity, or maximal reaction rate. This means it's not going to be active unless you have a lot of glucose, but if you do have a lot of glucose, it can act very quickly to phosphorylate it. Also, it has no feedback inhibition from glucose 6-phosphate, so as long as there's lots of glucose in the blood, glucokinase will actively phosphorylate it. You can think of this as acting as kind of a buffer system, because after you eat a large meal with lots of sugar, glucokinase will help take it out of the blood and store it in the liver and allow the beta cells to produce lots of insulin. Therefore, it also makes sense that insulin is not required for glucokinase to be active. Rather, glucokinase is used to get sugar into beta cells so they know to make insulin and secrete it. Then, this insulin is required for hexokinase in other cells to again phosphorylate glucose, which allows them to either store it or make energy. You can remember that glucokinase is found in the liver because both liver and gluco have 5 letters. The net reaction of glycolysis takes place in the cytoplasm without the requirement of oxygen, and is shown by this reaction here. You start with glucose, 2 inorganic phosphates and 2 ADP, as well as 2 NAD, and end up with 2 pyruvates, 2 ATP, 2 NADH, 2 hydrogens, and 2 waters. The important parts are that you need 1 glucose to make 2 pyruvates and 2 ATP, and in the process, 2 NADH molecules get reduced to 2 NADH molecules. Now let's go through the enzymes that regulate glycolysis one by one. The 2 steps of glycolysis that require ATP are the first step and the third step. First, either glucokinase or hexokinase uses 1 ATP to convert glucose to glucose 6-phosphate. As I mentioned before, glucose 6-phosphate has negative feedback on hexokinase, so when the cell has a lot of glucose 6-phosphate built up, it knows hexokinase shouldn't be working anymore. The phosphorylation of glucose effectively traps it inside the cell, because the phosphate group has a negative charge and therefore can't pass through the membrane. In the third step, phosphofructokinase 1 uses 1 ATP to convert fructose 6-phosphate to fructose 1,6-bisphosphate. This just means that it's fructose with phosphate molecules in the 1 and the 6 position. Phosphofructokinase is the rate-limiting enzyme of glycolysis, and therefore it can either be activated or inhibited by various substances. If cells have lots of AMP, or fructose 2,6-bisphosphate, this means that they don't have a lot of ATP and need to make more. So these both activate phosphofructokinase 1, which increases the rate of glycolysis. If lots of ATP and citrate are present, this means the cell has sufficient energy and does not need to continue glycolysis. Therefore, both of these inhibit phosphofructokinase 1 and slow the rate of glycolysis. There are 2 steps in glycolysis that produce ATP as well, but each one occurs twice, since they occur in the product of breaking fructose 1,6-bisphosphate into 2 separate 3-carbon molecules. This means that a total of 2 ATP are consumed and 4 ATP are produced, resulting in a net gain of 2 ATP, which I mentioned earlier. Phosphoglycerate kinase converts 1,3-bisphosphoglycerate into 3-phosphoglycerate. Judging by the names of the substrates, it makes sense that an ATP is produced, since 1,3-bisphosphoglycerate has 2 phosphate molecules on the 1 and the 3 position, whereas 3-phosphoglycerate only has 1, which is on the 3 position, which means that one of those phosphate molecules was taken off to make ATP. Finally, pyruvate kinase converts phosphoenol pyruvate into pyruvate and produces 1 ATP. This is activated by fructose 1,6-bisphosphate and inhibited by ATP and alanine, since again if you already have a lot of ATP, you don't want to keep going through glycolysis. Pyruvate dehydrogenase is the enzyme that links glycolysis, which takes place in the cytoplasm, to the TCA cycle, which occurs in the mitochondria. It decarboxylates pyruvate and adds the remaining acetyl group to coenzyme A, creating acetyl coenzyme A. This is inhibited by ATP, NADH, and acetyl coenzyme A. Since again this is a process used to make ATP, so if you already have it you don't want to make more, and also this produces acetyl coenzyme A, so again if you already have it you don't want to produce more. It's pretty logical to think why end products such as ATP can inhibit glycolysis, but the reasons why fructose 2,6-bisphosphate can activate some glycolysis enzymes may not be as intuitive. Fructose bisphosphatase 2 and phosphofructokinase 2 are actually part of one enzymatic complex, but respond in opposite ways to phosphorylation by protein kinase A. In the fasting state, high glucagon increases the amount of cyclic AMP through a G-protein coupled receptor system. This in turn increases the amount of protein kinase A, which phosphorylates and therefore activates fructose bisphosphatase 2 and inhibits phosphofructokinase 2. This means you're going to be converting fructose 2,6-bisphosphate into fructose 6-phosphate and not the other way around, which in turn allows gluconeogenesis to proceed to replenish blood glucose levels, which of course makes sense if you're in the fasting state. In the fed state, high insulin levels decrease cyclic AMP and therefore decrease protein kinase A. The fructose bisphosphatase 2 or phosphofructokinase 2 enzyme complex remains unphosphorylated, and therefore only the phosphofructokinase 2 portion is active. This means they'll be converting fructose 6-phosphate into fructose 2,6-bisphosphate, which activates phosphofructokinase 1, which allows glycolysis to proceed. Keep in mind that phosphofructokinase 1 is the rate-limiting step in glycolysis, and therefore it's important to regulate this enzyme more so than any others. The pyruvate dehydrogenase complex links glycolysis in the cytoplasm to the TCA cycle in the mitochondria. The net reaction converts pyruvate, NAD, and coenzyme A to acetyl-CoA, carbon dioxide, and NADH. The cofactors needed for this reaction include pyrophosphate, which is supplied by vitamin B1 or thiamine in the form of thiamine pyrophosphate, FAD, which is vitamin B2 or riboflavin, NAD, which is B3 or niacin, coenzyme A, which is B5 or pantothenate, and lipoic acid. The last one, lipoic acid, is inhibited by arsenic, in which case patients will have vomiting, rice water stools, and garlic breath. If you're exercising, you probably want to be undergoing aerobic metabolism, and therefore you want the pyruvate dehydrogenase complex to be active. The mechanism by which the pyruvate dehydrogenase complex becomes active during exercise is because the ratio of NAD+ to NADH is increased, the amount of ADP is increased, and the amount of calcium is increased. And these, in turn, can all activate the pyruvate dehydrogenase complex. The alpha-ketoglutarate dehydrogenase complex converts alpha-ketoglutarate to succinyl coenzyme A in the TCA cycle, and uses the exact same set of cofactors as the pyruvate dehydrogenase complex. So if you remember how one of these works, you can remember the other one as well, because they use the same cofactors and they both decarboxylate their substrates.